All right, everybody. Well, welcome to the very last day of August 2018. This is our August micro seminar. All right, everybody. Well, welcome to the, let's see, very last day of August 2018. This is our August micro. Sorry, everybody. I had the YouTube running at the same time, and I didn't realize it was going to click on like that. All right, so you don't have to listen to me twice, I promise. I am going to introduce you to our speaker today, which is, it's my pleasure to introduce you to Mario Toubes-Rodrigo, who's going to be speaking to us about glacial microbiology. We've actually had at least two glacial microbiology talks in the micro seminar. This will be a third, so we're developing a little bit of a subsystem here. So please feel free to look around and educate yourself on glacier microbiomes, because we're building quite a nice wealth of information on cutting edge research in this area. A little bit about Mario before he gets going. He did his undergraduate at Pablo de Olavide University in Seville. It was a bachelor's in biotechnology. He then did a master's in environmental micro at Autonomous University of Madrid, and then a PhD at the Manchester Metropolitan University, also in environmental micro. He's been teaching there at Manchester Met, and then he's going to be starting a postdoc in October at the Open University in the UK, and that'll be focused on astrobiology. And so we're excited to hear what Mario has to say about glacial microbiology in the middle of all these transitions in his career. So Mario, without any further ado, take it away.
Well, first of all, hello everyone. As Cameron has said, my name is Mario. Basically what I'm going to talk today is a bit about what I did on my PhD and give a bit of what I'm going to be doing in the nearest future. So if we start, I have a presentation here. Right, show. Should be working now. Can you see it?
Yes, just click your presenter tools.
Yeah. So I entitled this talk "Rolling in the Deep" because that's the part of the glacier I'm working in. I work on the subglacial part, so where the ice and the sediment meet. My main interest is knowing what kind of microbes live there and what they are doing. But let's get a bit more into that. So when people think of glaciers, normally they think of ice like this. So very nice ice, very blue, kind of bubbly. That's what we in glacial ice. And actually, every time I'm going to do some fieldwork, everyone is like, "Oh, Mario, do you mind bringing some ice for my whiskey?" And I'm like, "Yes, I could." But actually, what you should be expecting is something more like this. So this is basal ice. This is what the kind of ice I work with. And as you can see, it's quite muddy and quite dirty. But the most important thing is, yes?
I'm sorry to cut you off, but we're still just seeing it from the perspective of like your actual PowerPoint editing.
Okay, so give me a second.
And I think it'll be a lot prettier.
I think that what I need to do is the following, and now here. Ta-ta-dan, come on, let's do it. Is that better?
Yes.
Okay, so this is glacial ice, and this is basal ice. But I guess that the main question that we need to ask ourselves is, why is it important to study glaciers, and why is it important to study basal ice? Well, I guess that the main thing about glaciers is that if we think about how much land these glaciers are occupying, is that they occupy an 11% of the immersed land on Earth. That is a huge amount of land covered by ice. Nevertheless, we are not very sure of how they work and what is happening in them. Also, most of the research has been focused on the surface, and actually the first person to describe, and I'm going to put this in between commas, supraglacial microbiology was Aristotle, that talked about watermelon snow, which is basically snow that contains an algae that's going to make it go pink. But the important thing about basal ice is that it is the richest part of the glacial ecosystem. And until 1999, it was thought of lack of life. I was born in 1989, so actually the field I'm working in is younger than me. So microorganisms are known to play fundamental roles in different ecosystems. So this is not going to be an exception in a subglacial ecosystem. Microbes inhabiting basal ice are going to be playing very important roles that are going to have very deep implications. Finally, and as we all know, since the Little Ice Age, so since about the 19th century, glaciers have been receding. And nowadays, due to climate change, this melting on this receding is going even faster. So we don't know what kind of consequence the release of these microbes is going to have to the fallen once the glacier has melted. So this is, for example, the glaciers I've been working in. So this one here is Skaftafellsjökull, Svinafellsjökull, and Kuriajökull. So you can see that since the 19th century, they've receded quite significantly. So this is about a couple of kilometers. This is about 1 kilometer, and this again, a couple of kilometers. So another very important implication of subglacial microbiology is that actually we can think of basal ice as a very good analog for other planets. So actually, a couple of weeks ago, a paper was released in which apparently they found some subglacial water in Mars. So in the south polar cap of Mars, there is a 20-kilometer long lake. As we say sometimes in microbiology, if there's water, is potentially life as well. So the kind of subglacial environment that I am working with is going to be kind of similar to the ones we are going to have in Mars if there's life there. Also, we have Enceladus that has methane plumes. They've been trying to adjust the chemistry of Enceladus and the methane production, and it seems very uh, it seems that it could be quite likely that maybe there could be life in Enceladus producing this methane. Also, we have Europa, which also contains ice. And finally, one of my favorite things is that there is a period of time on Earth, the Proterozoic, in which basically the Earth was a massive snowball. So the only life that could thrive on those conditions was basically microbes that were adapted to cold temperatures, what we call psychrophiles. But why investigating basal ice instead of just getting into the subglacial environment itself? Well, because it's very hard to get into the subglacial environment. We need to think that glaciers can be quite thick in terms of how much ice they have. And actually, the last glacier I've been working in, Subglacial in Sweden, it's like 250 meters thick. So if you want to core to get to the bottom of it, it's going to be very hard. Nevertheless, we know that basal ice, this kind of very muddy ice I work with, forms underneath glaciers by different processes. So actually, it is a very good proxy to investigate how life could be underneath glaciers. So actually, in this picture here, this is a very happy Mario underneath a glacier like 2 weeks ago in Subglacial in Sweden. So, oh, what is this going? Okay, now here. When I first started my PhD, we had to decide what was going to be the question or what were going to be the questions that I was going to address. So the first one was trying to understand how the conditions for life underneath glaciers are. Secondly, if the life conditions for life were adequate, if there are going to be microbes there, and how these microbes are going to be. And finally, because of how oligotrophic, so how poor in nutrients this basal ice is, it's likely that the microbes are going to be interacting with minerals in the basal ice ecosystem. So we want to also understand how this interaction between mineral and microbes was going to be. So what kind of glaciers I've worked with? Well, here you have the three main glaciers I've been working on the right-hand side, Svinafellsjökull, Skaftafellsjökull, and Kuriajökull. And most of my PhD was focused on the first one, on Svina. And when I first started, I was very excited and I thought, oh my God, this glacier sounds so glamorous. But then I discovered that actually Svinafellsjökull mean the glacier of the hill of the peaks. So no glamour whatsoever, but still it's an amazing glacier. Quite likely you might have heard of it, maybe not the name, but you are very familiar with it because it has appeared in Game of Thrones. So basically I did my PhD Beyond the Wall. It has appeared in Interstellar, and it has also appeared in Batman Begins. So it's quite a cinematographic glacier. But why choosing these glaciers here? So first of all, they are temperate glaciers. As I always say, glaciologists have quite a twisted sense of humor because they call temperate glaciers to those ones that are around 0 degrees, and they refer to them as warm glaciers. Secondly, the bedrock of these glaciers, so the kind of substrate we have underneath the glaciers, is basaltic. Because if you think of Iceland, Iceland is a very volcanic island, and all you're going to have underneath is volcanic rock. And all of these glaciers over time have been carving basins underneath. Svina and Kyrajokul all of them are coming from the same big glacier, which is called Orifajokul. And then Skafta, apart from this glacier, is going to also derive partially from another glacier called Varnajokul. So there's going to be some difference between them that can have an impact on microbiology. So we wanted to analyze how these different conditions were going to affect the microbiology. So this picture here, this is a crevasse in Subglacial. This is like 10 meters tall. So I decided to creep underneath this crevasse to see if I could find any basal ice and to understand how this subglacial ecosystem was going to be. We can call it the basal ice or the basal ice ecosystem or the dirty and soggy bottom of glaciers and microbial ecosystem. So actually, on the left-hand side of the picture, there's like a whole eye cryptine, and I found this ice here. So you can see this, first of all, like a very nice transparent ice, but underneath you can see how sediment rich it can get. This is going to be the kind of ice I am interested in. The basal ice ecosystem is going to be dark. Obviously, not the part of basal ice is going to be exposed, but for most of the glacier, it's going to be very dark. It's going to be cold. It's going to be oligotrophic, and it's going to be very rich in minerals. So microbes surviving in this ecosystem need to be very adapted to these conditions in order to thrive. So this the basal ice is not just a single type of ice, but a group of different subtypes of ice that are going to form by different processes. So one of the questions that we asked ourselves was, is actually different types of basal ice going to harbor different microbiology? Is it going to be the same? So for that, we identified 3 types of ice in these 3 glaciers I've mentioned. The first one is going to be debris bands. Debris bands form in parts of the glacier where we're going to have a lot of pressure under the sediment and under the ice underneath the glacier. It's going to basically make it fall, and it's going to integrate part of this very sediment-rich substrate that we have underneath into the ice. So if we have a close-up look into these debris bands, we can see that basically we have a lot of sediment separated by clear ice. Second one is going to be dispersed. Some researchers have hypothesized that dispersed ice is going to come from these debris bands because as the glacier flows, the debris bands are going to be deformed and they're going to be kind of diluted into the rest of the ice, and it's going to generate structures like this. Finally, stratified is the richest in sediment ice that I analyzed, and it forms just at the end of the glacier where because of processes of freezing melting, we are going to have different layers of sediment into the ice, and it looks kind of like this. So now that we know what kind of ice we're going to be talking about, let's see how the chemical conditions of this ice is going to be and if it's going to be prone to have life. So chemically, we couldn't identify any differences between the basal ice and the basal ice types. But what we found most interesting is that we found that carbon, nitrogen, and sulfur are quite low. Maybe you can think that 1,000 ppm uh, um, carbon is quite a high amount of carbon, but you need to think that this is 0.1% of carbon in the sediment. This is the same kind of carbon concentration we are going to have in very hyper-arid deserts. So it's not very rich in carbon or nitrogen. However, the amount of iron that we have is rather significant. And iron has been related before with the survival of certain group of microbes that we're going to call chemolithotrophs, that actually what they can do is oxidize an iron to generate energy and fix carbon in order to generate organic matter. Also, what we found is that the ice itself and the sediment entrapped in the ice are going to be different. So whereas the ice is going to be richer in calcium and sodium, the sediment is going to be much richer in iron. And this is going to have some implications that I'm going to explain in subsequent slides. So also one of the things that we were most interested in is, right, so we have all these elements, but elements by themselves are not very indicative. We need to know what kind of minerals we are going to have. And for that, we've analyzed 60,000 individual grains of sediment entrapped in the ice. What we found is that basically the mineralogy of this sediment is going to be dominated by silicates, and silicates are going to be playing a very important role because some years ago, some people, so Tellin et al., discovered that actually silicates in subglacial environments when there's available water, as we have in temperate glaciers, is going to produce hydrogen. And this hydrogen can be utilized by some of the chemolithotrophic bacteria in order to generate energy. A closer look into what kind of minerals we have, we saw that the silicates were dominated by olivine and feldspar that are also known to harbor microbes. And also we have quite a lot of iron-rich and iron oxide minerals, such as martite or magnetite, that if you remember, as I just mentioned, they can be used by microorganisms. So we decided to count how many cells we have in the ice, and we decided to separate between the sediment itself, so the mud that we can see in the ice, and the ice matrix, so the clear ice that we have around the sediment. And what we found is that independently of the kind of ice that we were looking at, the sediment contains around 10 to the power of 7 cells, so that's a lot of cells, whereas the ice contains 2 orders of magnitude less. Based on this and knowing how what is the amount of sediment that is being released from Svinafellsjökull per year, we calculated for the first time how many microbes are escaping per year from an individual glacier. And the number is quite overwhelming. We are talking about 10 to the power of 16 cells per year, escaping from the basal ice layer into the margin. And we have this huge amount of cells, but we don't really know what they are doing. Well, now we know, but I am not going to spoil it because obviously we have this talk for something. So the next thing that we did, we know that we have the conditions for life and we know that we can see cells. So we want to know what kind of cells we have in this subglacial environment. So for that, we decided to analyze the microbial diversity to a phylum level, which is basically like saying mammals or fish. So it doesn't give us quite a lot of information. We found that basically the basal ice ecosystem is dominated by proteobacteria, which is not surprising because if we see the ice, it's kind of similar to soil, and proteobacteria are quite dominating in soil. However, if we decide to go to genus level, we're going to see something very interesting because when we resolve to a genus level, we can actually kind of predict what kind of functionality these microorganisms are going to play in the basal ice ecosystem. And we found that the main group is Lysobacter. Lysobacter is a heterotrophic microbe, so it's a microbe that feeds on organic carbon. It can't fix carbon itself. But the most interesting thing about this microbe is that it contains a very powerful battery of enzymes. So this battery of enzymes can break loads of different carbon sources. So if we have cells that are dying, the carbon that we're going to have in these cells is going to be very complex, and therefore normal microorganisms won't be able to use it. However, Lysobacter has the ability to actually break this very complex carbon, making it available for all the microbes to use it. The second and third groups of genera that we identified was Polaromonas and Thiobacillus. Both of these genera are known to have chemolithotropes, but they actually have different capabilities. Whereas Polaromonas is able to oxidize hydrogen, Thiobacillus can either oxidize iron or sulfur. Then Methylotenera can also use carbon C1 compounds, so very simple carbon sources to generate organic matter. Gallionella can oxidize iron again. So we can see that actually we have a very significant amount of chemolithotrophic bacteria that are going to be able to oxidize different elements in the basal ice to generate this organic matter and potentially sustain an active ecosystem. But the next thing that we wanted to do is, okay, we have all these potential predictions just looking at genera and what they do about how the ecosystem can be. But actually what we can do is we have tools nowadays in which, based on the diversity of microbes that we have inhabiting an ecosystem, we can try to predict what kind of functionalities we have. And actually, on the figure on the left-hand side, we can see that the majority, the majority of potential functionalities are related to metabolism. And a significant part of it actually is going to be energy metabolism, because this ecosystem is going to be quite poor in energy. So they need to find ways to generate their own energy. So what we found is actually, if we have a image, if we have a look at the second image, we have oxidative phosphorylation. So basically, that's production of energy is the most energy yielding process. But we have methane metabolism. Methane can be it's a very concerning greenhouse gas nowadays, because if we think of the potential of carbon dioxide for climate change, we need to think that methane actually has 23 times more greenhouse power than carbon dioxide. By methane metabolism, we not only are talking about production of methane, but also potentially all the microbes can oxidize methane to generate another gas that are going to be less dangerous. And actually, on the next slide, I will talk more about this kind of microbes. The third most important group, or second, depending on how we look at it, is carbon fixation in prokaryotes. So basically, these microbes that can oxidize minerals or elements in order to generate energy and fix carbon. And if we compare it to potential photosynthetic fixation, we see that chemolithotrophic metabolisms are going to be much more abundant than phototrophy. That's not surprising. I mean, I've mentioned that this basal ice is a rather dark environment. But what is surprising is still there's some potential phototrophy there. And what is more interesting about this is, if we have a look at the previous slide that I showed, we have a very small amount of cyanobacteria here. Cyanobacteria can actually use the energy of sun, they are photosynthetic microorganisms, to generate energy. What is going to be most interesting, not in the basal ice itself, but as we mentioned, potentially due to this recession of glaciers, these cyanobacteria are going to escape to the foreland, and maybe they can reactivate there, and they can start fixing carbon and enriching the sediment, making it more prone to the colonization of plants, for example. Also, we have some minor metabolisms here, like sulfur of nitrogen. But as we mentioned in the chemical composition of the sediment, these elements are going to be very low. And actually, if we compare nitrogen and sulfur metabolisms to either carbon fixation or oxidative phosphorylation are going to be very minor. So with the information of the different taxa, the different groups of microbes that we found, we can build what we call a network analysis. So basically, what this figure on the left is showing is how microbes statistically are interacting with each other. And one of the most relevant things I would say is that if we look at anything that is not pale yellow, they are generally very related to other microbes. This is very interesting because we have these microbes that can oxidize minerals or elements, and they are fixing carbon, and then other microbes can fit on this fixed carbon. So this is why they are going to be interacting so close to each other in what we call biofilms. So basically, it's like a family dinner in which these chemolithotrophic bacteria are going to make in dinner, and the rest around them are going to be using this carbon that they fix in order to thrive in this very carbon poor ecosystem. The figure on the right is based on the groups that we know, how um, how important the different metabolisms in the different ice taps is going to be. And what we found is actually that the oxidation of iron, sorry, is going to be significantly more abundant in stratified ice, whereas hydrogen oxidation is going to be more significant in dispersed. The theory that we have behind this is that stratified is newer, and I'm going to put this between air commas, in terms of formation of basal ice. So the stratified ice is going to form near the end of the glacier. And the iron contained in this stratified ice is likely to be more reduced. So then microbes can oxidize it. However, as we mentioned before, the dispersed ice proceeds from debris bands that form in the beginning of the glacier, and as it flows, is going to form this dispersed ice. During this process, if the sediment has been colonized by microbes, the microbes are going to be using the iron containing in the ice, and they are going to be exhausting it over time. And that's going to reach a point in there's not going to be more reduced iron, so then they need other sources in order to survive. And one of these is going to be the silicates that are going to be generating hydrogen, because actually this reaction is very slow in time. And actually, also, if you remember, we have a lot of silicates, so it is not likely that this hydrogen is going to exhaust as soon as the iron. So with all this information and these bits and bits, we decided to generate some kind of model of how this ecosystem is going to work. On the top left, we have, for example, a particle that is going to contain iron, and it's going to be colonized by these iron oxidizing bacteria that are going to be oxidizing it and generating nutrients that heterotrophic bacteria are going to use. On the right-hand side, for example, we can have a silicate that's going to generate hydrogen. And I'm going to go back to the previous slide because there is something I want to highlight. And is this very small group here contains these two purple groups here that are going to be methanolinia and methanospirillum. These two groups are methanogenic archaea. So methanogenic archaea are a group of microbes. They're not bacteria, they're slightly different, that can generate methane. And actually, methane from a biological perspective can only be generated by archaea. So what these microbes can use is using hydrogen and using carbon dioxide in order to generate this methane to get energy as other chemolithotropes will do. And also, if you remember from the previous slide, I mentioned that we have quite a lot of methane metabolism. And actually, we have groups here, such as methylotenera or methylibium, that is 3A, so it's somewhere around here, 3A here, that actually what they can do is utilizing this methane, oxidizing it to carbon dioxide to get energy. So maybe there's some kind of balance, but we don't know so far. So we don't know if actually the recession of these gases are going to be releasing this this methane to the atmosphere. It all depends on how active the methanogenic archaea and the methanotrophic bacteria are going to be. So yeah, we have, as we mentioned before, we have the hydrogen oxidizers and the methanogenic archaea here that are going to using the hydrogen generated by silicates to generate organic carbon. And potentially, the hydrogen can become methane in the presence of methanogenic archaea. Also, although we didn't identify any sulfates per se using Raman, but we saw some using the single particle analysis. Um, Sulfates can also be generated by microbes, but it is quite unlikely in this environment regarding that we don't have that much sulfates, that much sulfur. And finally, we also have carbonates here that actually carbonates in the presence of acids that microbes can produce can be dissolved, and the elements that can be quite limiting in an ecosystem such as basal, such as iron or potassium or phosphorus or sodium, can actually be released from the mineral matrix into the ice. Also, what this figure is showing here is that we can think of basal ice as a block of ice that is going to be completely hermetic. Actually, what we find is that basal ice is going to be made of small crystals of ice, and between these ice crystals, we are going to have liquid water. This is also going to happen around the grains because due to physical interaction, there is always going to be some liquid water around the minerals. And actually, this is what is going to explain why we have a higher number of microbes in sediment than the one we are going to have in the matrix, because this sediment, these grains of sediment we have in the matrix are going to be feeding microbes, and they're going to proliferate much faster around these grains that if we have some microbes around here. Also, what is very interesting about this kind of environment is if we think of salt and ice, we know that salt is going to dissolve ice. But what happens when ice forms in the subglacial environment is that we're going to have some solutes, we're going to have some salts. And as the ice forms, the ice is not going to entrap salts. The actual salts are going to be concentrated in these veins around here that are going to make them very, 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 very rich in salts. So actually, it can be quite a harsh ecosystem for microbes due to these very salty conditions. So, what are the main roles of microbes then? What kind of effects these microorganisms are going to be playing in basal ice? So, first of all, we are going to have the microbes are going to be enhancing weathering, because as we said, they can interact with the minerals that they can oxidize them. So that's going to enhance weathering. And the picture I'm showing here is not a picture of mine, is blood falls, which is a very saline ecosystem under a glacier in Antarctica. And it's very rich in iron. Because of the activity of microbes, it's very anoxic. So the iron is going to be reduced. And when iron is reduced, it doesn't have much of a color. But there was a breach in this ecosystem. The water started escaping, and when it became in contact with the air, it very rapidly oxidized, and this is why it has this very nice red color. Secondly, we have that microbes in the ice are likely to be producing gases. It can be methane, as we mentioned, or it can be carbon dioxide. And these gases are going to have a very relevant role in global warming because they have greenhouse effect. We don't really know what the implications of the release of these gases is going to have, but it is likely that it's going to present some kind of positive feedback loop in which the more the glacier is melting, the more gas is going to escape to the atmosphere, and the more gas is going to escape, the warmer is going to get, and therefore the more the glaciers are going to melt. Finally, this is soil. Microbes can also enhance soil formation, and this is something that is not new. It is something that we had known for years. That once that we have a deglaciated ecosystem, the microbes are going to basically weather the glaciated sediment to generate soil. What microbes can do is basically fixing carbon, enriching the carbon, and then they can also, once it is reaching carbon, they can also fix nitrogen. And then once this sediment is going to be rich in nitrogen, then other organisms, such as plants or lichens, can colonize them. However, the most relevant outcome from my research is that now we know how many microbes are escaping per year from this glacier, from Svina. And now we also know what kind of potential activities these microbes have. So we've talked that we have quite a lot of iron and we have silicates. So the microbes can actually oxidize this iron and the hydrogen from the silicates, enriching the amount of carbon in the sediment. And then what we can have is that there can be a deposition of microbes that have been swept in by the wind. And if we have cyanobacteria or any other nitrogen-fixing bacteria, then this nitrogen content in the sediment is going to increase, and then all the microorganisms will be able to settle. So for me, the take-home messages from this talk that I've just given is that basal ice is an oligotrophic environment that contains minerals that can be used by chemolithotrophs, especially iron-rich minerals and silicates. Secondly, that the basal subglacial environment is releasing a huge amount of microbes that can generate soil. And finally, that the basal ice microbiology is dominated by chemolithotrophic bacteria, especially iron oxidizers and hydrogenotrophic bacteria. So that's what I've done on my PhD, but my research doesn't end there, and I have some future plans. So as I just mentioned before, I've come from Sweden last week, in which actually what we're trying to do is analyze the microbiota of certain glaciers. So the first one that we have here, sorry for that, first one here is Subglacial, this one here. Basically, Subglacial is to glaciology what E. coli is to microbiology. It's the most studied glacier ever. Then we have another glacier here. This is Subglacial. You can't see it here, but there's another very pretty glacier here called Kebnepakte-Glaciaren. These glaciers are very different to the ones that we studied in Iceland. These are cold-based glaciers. Presumably, the microbiota is going to be different, but until we don't analyze the entrapped DNA, we won't be able to say it. Also, as Cameron has just mentioned, I am going to be studying a postdoc in October at the Open University, in which we're going to be analyzing the astrobiological implications of sulfate-rich lakes. This is all from my side. I just want to thank my PhD supervisors, David, Robin, and Simon. This is Joe. This is one of our colleagues. And this is my last field trip to Iceland in 2016. And then some other people such as Eda, Sania, Jessica, Trish for all the help. And also the Royal Geographical Society and the Manchester Geographical Society for the support of my research. I think that's all from my side. So back to you, Cameron.
Okay. Well, thank you very much, Mario. That was illuminating. Some really beautiful images in there too, along with the microbiology. let's see. We only have one other person with us in the seminar, so if you'd like to ask a question, go for it. If anybody is out there watching on YouTube, you can either tweet your questions to the hashtag #USeminar or to @microseminar. And while we're waiting to see if anybody else has questions, I have a couple. So, you know, there's a lot of really interesting evidence here for the chemolithotrophic metabolism. Are you planning on measuring chemolithotrophic activity at any point in the future?
I would love to, and that's on the cards, but it depends on how we can get the funding to do it. However, what we actually found is by culturing, we've actually isolated ion oxidizing bacteria. However, and this is one of the anecdotes of my PhD. First of all, psychophilic bacteria are very slow growers.
Right.
So even heterotrophs can take weeks until they grow. So once I was back from my last field trip, I just plated some of the samples in order to try to get if we had any ion oxidizers. The day before finishing my PhD and the deadline for my submission deadline, I checked on the plate that nothing had grown before, and I found that they had grown.
Okay.
But obviously, it was far too late, so I didn't have the time to sequence them whatsoever. But I know that they are there. I know they are ion oxidizers because it's quite a restrictive medium to start with.
Yeah.
And actually, you can see that inside of the colonies, you have red because they are oxidizing the ion and precipitating inside of the colony.
Okay. Mario, can you share your screen slightly differently so that people don't have to stare at me?
Give me a second. Is that me? Yes.
There we go. Now we can see you again. Good. Yeah. Okay. So you do have some iron oxidizing isolates out of here. That's great. So there seems to be. I mean, I guess I noticed also in your metagenomic data that there was high incidence of you know, basically aerobic metabolism genes. So do you think that the system is mostly like, or is the ice like? Is there any? I mean, I know in sediments things can go anoxic pretty quickly, but so is the ice permeated by oxygen like at all levels, or are there anaerobic zones, or, you know, especially in that like matrix you were showing as taxa proliferate in those little tiny spaces, do they ever go anaerobic, or do you ever see anaerobic taxa popping up in these situations? Besides, obviously you've got the methanogens, so there's got to be some way, but I'm wondering about other anaerobic metabolisms and sort of the interplay of oxygen in this whole system, basically.
Yeah, there's some anaerobic taxa in among our data, and actually previous literature has also described that there are anaerobic pockets within the basal ice. So it is likely that depending on the part of the ice that you are at, you're going to have either aerobic or anaerobic metabolisms. Okay. And actually, what we found as well is in some of the analysis that there's a very strong correlation between some aerobes and some anaerobes, and then some chemolithotrophs. So it seems that they might be building a biofilm in which you have the chemolithotrophic species in the middle, then the anaerobic species, and then the aerobic species. But this is just based on predictions. We actually haven't been able to actually put a probe to see what the redox potential of the different pockets are. Because actually in ice is quite hard because it's a solid matrix.
Right. Yeah. Yeah. Well, it's interesting to think about the structure of the actual organismal groups that you're finding in there. Are you thinking about testing any of those network predictions by like looking at fish or something like that to try and see if those organisms are in close association?
That's on the cards as well. And actually, one of the things that I was asked when I went to Sweden the last time is if I could bring some samples in order to analyze for further analysis. If we could do any further analysis. So I've extracted the DNA now. It's just ready to send to be sequenced. But I still have some pristine ice that we are going to check for. We can do fish, and we also want to do some extra culturing. So it's all on the cards, but we still have to decide what to do and what not to do.
That's great. Yeah. Well, let's see. So I don't have any other questions that have come in, but if someone wants to, let's say someone's watched this a few weeks from now and they want to get in touch with you right now to ask you some questions, what's the best way to get in touch with you?
By Twitter, probably.
Okay.
Because at the moment, my email address, I don't know what is going to happen with my email address because I'm changing of institution.
Of course. Yeah, and so your Twitter address is at @micro_Mario, correct?
Yeah, that's it.
Okay, great. Well, thank you very much, Mario. Appreciate your time and for contributing to the seminar. This is great. Thank you.
Cool. Thank you for inviting me. And you're doing such an amazing job. I really enjoy the micro seminars, in all fairness. So
Great.
it's great to be here.
Yeah, well, great. Best of luck in your postdoc. Hopefully we'll see you again.
Oh, yeah. You're not going to get rid of me that easily.
Okay. Well, take care, and thank you very much.
Bye.